could be instrumental in redefining the relationship that community projects have to sequencing centers. We would need to have much more integrated tracking systems. The EMP has a mechanism for tracking samples and so forth, and we want to find a way to have high bandwidth integration with the similar systems at the sequencing center, so that it's sort of like buying something on Amazon. You can see exactly where you are all the time. And we would want that between the EMP systems and whatever systems could exist out there in the sequencing centers. And that means transacting with these sequencing centers would be quite different. It would be like having an internal partner as an interface. And these pilot projects could help define that. And this could really revolutionize how business is done with sequencing centers, perhaps. And if we have various kinds of service partners in this project that want to sign up for different parts of the task, high throughput sample prep, for example, done separately from some place that might be doing the sequencing, this kind of high bandwidth interface between that kind of service operation and the sequencing center would be necessary. This is done in other kinds of industry all the time, but it's not done yet in sequencing. Now, what could really change our assumptions in this whole model? Well, right now we don't have technology to directly interrogate complex microbial environments. We could imagine in our mind's eye maybe some technology that would allow us to do geochemical environmental mapping down at the sort of micron level, or maybe 100 nanometer level. Some kind of magic tech that would give us a very detailed picture of an environment and maybe directly able to access the diversity of that environment. If we had that, right, we'd be crazy to try to infer the structure of these communities rather than just directly read it. Of course, we don't have that, but maybe someday we will. If we can directly access environmental metabolomics or proteomics data, we want to add that to the mix. Those would become very important types of data that would help us and maybe reduce the amount of sequencing we would need of genomes, or maybe not, it would increase it, but it would complement what we're doing. And of course, we spend a lot of time, especially late at night in the bars, talking about, well, it'd be great to have a robot that we could send off and collect samples, or maybe to collect the sample, do the sequencing, and just radio back the sequence, right? Because right now it's actually a major difficulty to get people out into the field, particularly in places that are remote. So those things could change things. If we had a dramatic improvement in the ability to do microcosm experiments, that would also change this, right? You could construct experiments to answer some of the questions that we're trying to infer indirectly through the environmental sequencing, then we could be more targeted, right? If we had reliable theories about how artificial microbial communities were constructed and ways to do time-dependent measurements, then we would, again, adjust our strategy. Right now, almost all these things are wish list items, some of them completely crazy. But the point is that some of this may happen, and this project would need to adjust itself in that context. So what's actually happening in the sort of phasing of this? Well, the project sort of got created in the summer of 2010, and what we imagine doing in the rest of this year is getting more headway on the standards. We have, I would say, draft or proposed standards now. There's been some starting of the pilot, with samples coming in from around the world to the handful of labs. We built a prototype of this environmental sample database that is available for tracking things. I think it probably needs more work. But to launch the pilot project, or to push it more forward, that's targeting about 10,000 samples, in the next 12 to 18 months, that would be collected, acquired, processed, and so forth, and sequenced in a set of centers that are associated with the bootstrap of the project. And we hope very much that our colleagues here today are a part of this. 2012 and beyond, our plan is to, and this is a sketch of what we hope to do is to have a series of these biological driver projects sort of flooding the public opportunities for access to sequencing. All of them specifying in their request to use the EMP standards and protocols. So we can try to develop some momentum around this idea of a research proposal agreeing to do a certain set of standardized steps that would allow the data to have more value than individual study. And to use the results of the pilot in 2012 demonstrate that by doing this, we can do analysis runs within and between driver projects that are comparable in quality. So I want to thank the folks who've been really driving this. I sort of feel like I'm the, I don't know, the crazy uncle that tries to keep nudging everybody forward, but certainly Jack and Volker, Rob and Janet and Jed and Bindu and Mark, Jonathan are really intellectually pushing this forward. And it's without the energy that comes from this group and the sort of incredible amount of creativity, I don't think this would be happening. I just realized that I don't have Nikos' name on here. Nikos, I'm sorry, you're a part of this crew. And so I want to thank them. They're really the ones that you should grab. And certainly if you don't like this idea, you should punch them first before you talk to me. So that's my intro. I'm happy to try to answer a couple of questions before we go.
Sharing and the sustainability. We need the money, but we also have raised that banner of free sharing of the genome upstream data. That's because. Thank you.
So I think this is a very good point. The project that's most similar to this, at least in its origin, is the Human Microbiome Project in terms of how it got started in France 5 or 6 years ago. And I would say it is stumbling on the data sharing problem. One of the reasons that we like having Jonathan on this list is Jonathan is a huge advocate for open access, both for results and the data. And I want to note with great appreciation that VGI released the E. coli data just a few days ago with the Creative Commons level 0 license. I think personally that's the right way to go. That we're entering a new way of doing science. There's going to be so much data that the groups that can be comfortable with pushing the data out immediately, even if it's flawed, right, even as they're correcting it for the next generation, will capture the imagination of the community because that they will be most responsible for accelerating the science. So I personally believe that the data for this must be open, should be openly available from the instant it comes off the machines. I mean, as soon as you do your minimal quality control, make sure it's not garbage. And that the mechanism for, we're going to have to invent a new way for people to identify themselves with the scientific output. And I can't say for sure how we're going to do that, but I think I want to give VGI a round of applause for releasing the E. coli data that way. I think it's the way to the future.
Okay, thank you, Mr. Stevens. And we will start our section. So the next section will